Welcome back to class. This is section 18.2, Archaebacteria and Eubacteria. Let us begin. So, Archaebacteria and Euchebacteria. What? Archaebacteria and Eubacteria, sorry. I had a brain fart. You guys probably heard the word bacteria. Most of the bacteria that we know about are going to be these bottom ones right here, this Eubacteria. Archaebacteria, some of you might know. But these are the strange guys. Even the book, we only get like a couple paragraphs on them, and then we switch over to them because these are the ones that you will interact with a lot more in your life. So we got to remember a couple vocabulary words, prokaryotes and eukaryotes. Recall that prokaryotes are unicellular organisms that do not have a nucleus or membrane-bound organelles. Okay? So prokaryotes do not have a nucleus, and they do not have membranes around their organelles. We do. We are eukaryotes, but these are prokaryotes. There's two kingdoms of them, essentially, two big chunks of them. There's the Archaebacteria and the Eubacteria. There's actually quite a bit of difference between these two, which is why they get their two own families. But they still have some similar characteristics that we put them in the same section for this purpose, for the purposes of this book. So here we see a couple pictures. There are three types of Archaebacteria that live mainly in extreme habitats where there is usually no free oxygen available. You can see some of these environments in this picture right here. One type of Archaebacterium lives in oxygen-free environments and produces methane gas. These methane-producing Archaebacteria live in marshes, in the bottom of lake sediments, and in the digestive tracts of some mammals such as cows. So some of these crazy bacteria live inside of the cows. They are found at sewage disposal plants where they play a role in the breakdown of sewage. We also have some of these Archaebacterium that live only in places with crazy amounts of salt, like Great Salt Lake in Utah, or the Dead Sea in the Middle East. There's even some that live in hot acidic waters of sulfur springs. They can live like on the bottom of the seafloor where you see those sea vents coming out and shooting up all that hot, scalding water. So they can live down there. So that's pretty much all we get about these Archaebacteria. They live in the crazy parts of the world that you wouldn't think anything else lives. These guys can make it there. They're specialized for there. They actually can't really live anywhere else but the places that they're specialized for, which is pretty interesting. So that's it for the Archaebacteria. Now let's spend the rest of this section learning about the Eubacteria. Eubacteria, which is the other kingdom of the prokaryotes, includes those prokaryotes that live in places more hospitable than Archaebacteria inhabit, and that vary in nutritional needs. There's three categories that we're going to look at real quick. We're going to look at the heterotrophs, the photosynthetic autotrophs, and the chemosynthetic autotrophs. So first we have to remember, what does heterotroph mean? Hopefully we remember. That means the organism needs to eat something to get its food. It does not make its own food. Okay? So here we see the heterotrophic Eubacteria live almost everywhere and use organic molecules as their food source. So almost anywhere you can think of, there's some of these guys on that surface. There's some Eubacteria somewhere. Some bacterial heterotrophs are parasites, which means they hurt other organisms. They are not adapting for trapping food that contains organic molecules or for making organic molecules themselves. Other Eubacteria that are heterotrophs feed on dead organisms or organic wastes. They're also called saprophytes. Remember those saprophytes. Saprophytes. Those also break down. Those are the ones that break down the dead organisms. Those are the names of those. Next, we got photosynthetic autotrophs. So hopefully you guys can kind of put this together. Hopefully remember autotrophs mean they make their own food. And if your next question is, well, how do they make their own food? Well, this word right here should tell you photosynthetic autotrophs. So they undergo photosynthesis. They need sunlight because they need to undergo photosynthesis. Here we see cyanobacteria are photosynthetic autotrophs right here. They contain chlorophyll just like plants do. They're kind of blue, kind of green, just like plants. I know plants are mostly green, but some plants have a little bit of bluish green to them, and some have red and yellow. Sorry, not plants. Some of these guys have a bluish green color to them. and some are red and some are yellow because of different pigments. But they all have the pigments because they undergo photosynthesis. They commonly lived in ponds and streams, moist areas of land, anywhere there's pretty much water. These guys are going to chill in there. And you can see they're attached in like these little chains. That's just how they hang out. Then we also got these weird guys, chemosynthetic autotrophs. So once again, autotrophs just means they make their own food. Chemosynthetic means they use chemicals to make their own food. The chemosynthetic bacteria do not obtain the energy they need to make food from sunlight. Instead, they break down and release the energy of inorganic compounds containing sulfur and nitrogen. Some of these are incredibly useful because they help produce nitrogen compounds that plants need. So these guys can use inorganic compounds to make compounds that plants can use, that we can eat, that we can use. So these guys are really important. So let's look at the structure of one of these bacteria. Prokaryotic cells have ribosomes, but their ribosomes are smaller than those of eukaryotes. Remember, prokaryotic are these guys, and you are eukaryotic. So their ribosomes are smaller than yours. They also have genes that are located for the most part in a single circular chromosome. Here it's kind of folded around, rather than pairs of chromosomes. We see that they have a cell wall. The cell wall is interesting because most of these bacteria live in watery environments. If we think back to osmosis, we remember that if the inside of the cell does not have as much water concentration as the outside, then the water will try to enter the cell. And if it does that too much, the cell will explode. Bacteria do not want to explode, so what stops them from exploding is this cell wall. It essentially constricts the cell to make sure it can only be this big. So if water tries to come in, it can't expand the cell enough to blow it up. Here we see the bacteria has a flagellum to help move it around. We learned about those a long time ago. Most of the bacterial genes are in here, but some of them are in plasmids. They're extra little chunks of chromosomes out there. Just like every other cell, there is a plasma membrane right here. It's a plasma membrane, cell wall, and then a capsule. Not all of them, but some bacteria have the capsule. And we see here a bacterium with a capsule is more likely to cause the disease than bacteria without a capsule. So that's something we have learned. There's a couple ways to identify bacteria. The book explains something called a Gram stain. So essentially we put this stuff on these cells, and depending on what these cells are made out of, the bacteria are made out of, they respond in two different ways. After staining, gram-positive bacteria are purple, and gram-negative bacteria are pink. Okay, so this stain stains them either purple or pink. The interesting thing about that is the reason why they get those colors is because of their structure. And that also gives us an insight as to what type of antibiotics or what type of antibacterials will help fight these different types of bacteria, whether or not it's pink or purple based off of this gram stain. We can also help identify bacteria by using different shapes. We got spheres, we got rods, we got spirals. Some bacteria like to hang out in pairs, some like to be in chains. So we name them essentially based off of different root words, like we do a lot of other things in science. We see here that if the bacteria has something called diplo in front of it, that means it's a paired arrangement of cell growth. If we see something like staphylo, that describes an arrangement of cells that resemble grapes. If we see something like strepto, that's a prefix that refers to an arrangement of chains of cells. So each thing about the names of the cells help us already to determine what kind of bacteria it is, assuming we know what all of the prefixes and suffixes are already. As I said, we're just kind of going base level introductory into this kind of stuff, so we won't go over all of them. But on the left, we see Streptococcus pneumonia, which a bacteria that causes pneumonia. I'm pretty sure I pronounced that last word wrong, but I know this is pneumonia. This might be pronounced a little differently. We have Bacillus anthracis, which causes anthrax. You guys are probably a little too young, but in 2000-ish, 2001, terrorists were sending envelopes full of these bacteria to infect people, and that was a weird time. I'm not sure what these guys do, Spirillum volutans, but they're a bunch of little spirals. You can't really tell in this picture. So bacteria do reproduce. They don't undergo mitosis, but they kind of undergo something similar. It's called binary fission, and this is how they produce asexually, which means they're just duplicating themselves. They're not exchanging genetic information with anybody or anything. To reproduce this way, bacterium first copies its chromosome. Then the original chromosome and the copy become attached to the cell's plasma membrane for a while. The cell grows larger, and eventually the two chromosomes separate and move to opposite ends of the cell. Then a little partition forms, and then eventually that separates into two separate bacteria. So there's not as many steps as mitosis. That's why it's not technically mitosis, but you can kind of see the resemblance. The chromosomes are duplicated, and then they kind of move to the opposite sides, and then eventually it splits right there. In a perfect world, some bacteria can reproduce every 20 minutes, which is terrifying, until you realize that bacteria can't reproduce that fast all the time. If they did, bacteria would cover the Earth's surface in a matter of weeks. Cover the Earth's surface in a matter of weeks. But they can't always be doing that because they might run out of food, or they might run out of water, or bacteria also produce waste, and if that many are around in one spot, then their waste will actually kill themselves. They'll be swimming in their own waste, which is disgusting. And things eat bacteria also, so if things are eating the bacteria, they're keeping the numbers down as well. Some bacteria, though, can undergo sexual reproduction, which means they do exchange genetic information with another bacteria, and that's called conjugation. And here, we see genetic material being transferred right here through the process of conjugation. During conjugation, one bacterium transfers all or part of its chromosome to another cell through or on a bridge-like structure called a pilus. Just like makes a little tube and sends the DNA in there, or on there, either way. But that way the new bacteria does have a new combination of genes that might be better adapted to survive. Speaking of surviving, you know that you need oxygen to breathe. However, not all of these organisms, not all these bacteria need oxygen. So recall that breaking down food to release energy is called cellular respiration. Modern bacteria have diverse types of respiration. So many bacteria require oxygen for respiration. These are called obligate aerobes. All right? So the way I remember this is they're obligated to use air, so they're obligated to use oxygen. They need oxygen. The organism that causes tuberculosis is one of these examples. And there are other bacteria called obligate anaerobes that are killed by oxygen. So if these guys, if the anaerobes, and meaning against, essentially, against oxygen, if the obligate anaerobes come in contact with oxygen, the oxygen can kill them. There's a couple different examples. Here we see the bacteria that causes this STD is an obligate anaerobe. Food poisoning bacteria is also the obligate anaerobes, when we're talking about botulism specifically. And then, aside from these two things, that oxygen can either be required or kill the bacteria, sometimes bacteria are out there chilling, infecting things, but then they say, you know what, the environment's a little too sketchy right now. I gotta hibernate. Hibernate's not the word you use for bacteria, but if they ever are faced with unfavorable environmental conditions, the bacteria can produce endospores. Okay? What endospores are the bacteria essentially takes its DNA, takes a little bit of the cell jelly, the cytoplasm, makes a little coating around it that resists drying out, and kind of that's the end of it. That makes a spore. So it's just a little bit of cell juice and the DNA inside of it, and that will wait for more favorable conditions to become an actual bacteria later. The crazy thing about endospores, they can resist drying out. So if it's super dry, the bacteria will still be alive. They can resist temperature changes, so if it gets a little too hot, they're fine. If it gets a little too cold, they're still fine. They can resist some chemicals, so you try to clean it with antibiotics or whatever, and the endospore still might survive. And get this, Some endospores have germinated after thousands of years in the resting state. So there could be bacteria right now that exists as an endospore and it became an endospore thousands of years ago. That's crazy. That is insane. Just think of if there's any bacteria hiding out in any of the melting ice caps or glaciers that have been an endospore for thousands of years and now they're being released. I know that's like a common like sci-fi writing prompt right there. If the bacteria is chilling like this for thousands of years and global warming melts it, then it's released, then what happens? Like good science fiction, I think, is always rooted in some type of real science. So endospores, you can imagine, are pretty useful for bacteria because they allow them to survive. For humans, though, we don't really want that, which is why there's certain rules to how we cook food and how we store food. So here, for example, canned food. Commercially canned foods have to be heated to at least 121 degrees Celsius for about 20 minutes to make sure that we kill all of those spores. It's not enough just to put them in boiling water. Boiling water does not get rid of endospores. If these foods are improperly canned, or someone does it wrong, essentially, what can happen is the obligate anaerobes, or the things that don't want oxygen, kind of wake up inside of that can, because inside of that can there's no oxygen, right? So all of the obligate anaerobes are like, wow, this is perfect conditions, no oxygen and a bunch of food, I'm just going to start eating all this food. And the bacteria eats and eats and eats this food inside of this can that was improperly canned, and they produce toxins. Well, if we eat those toxins, that could be bad. It can get us sick, or in some cases, it can even kill. We see right here a disease called botulism. Although rare, it can be fatal. And even potatoes kind of have this, which hurts me to the soul, to the core. If a potato is eaten immediately, the spores cannot germinate. However, if you bake a potato and you let the wrapper cool down into room temperature, and you leave it wrapped up in this, well, that creates an anaerobic environment, so that wakes up all of the bad bacteria, and then they will start to reproduce and produce the toxin. So baked potatoes, good, but baked potato and then cool it down, and then eat it after, not so good. Maybe not do that. Once again, we see anthrax right here. Bacillus anthracis lives in the soil. If it ever infects like your hands, it's not that big of a deal, but if you inhale it, then it can cause an infection in your lungs, and that can be fatal. And that's what people were doing, that's what some terrorists were using in the early 2000s to spread that disease to a couple of important people in the American government. Crazy times, crazy times. So up until now, we've kind of focused on the bad things of bacteria, or how things can go wrong. But bacteria are important for us as a species to survive. The disease-causing bacteria, yes, they exist. They definitely are out there, and we do want to get rid of them, or at least be able to fight them once we get infected. But there's other reasons why we need bacteria. For example, nitrogen fixation. So we kind of already talked about this. There's a lot of nitrogen in the air, but you can't use it because the air is 70% nitrogen, and you can't use anything. Or 75, I think, 77. Oh, this is 80, so it must be close to like 77 or 78% of the atmosphere is nitrogen, but you can't use it. Plants can't use it. Who can, though? There are some bacteria. Some of the bacteria can take the nitrogen out of the air, undergo nitrogen fixation, and change that nitrogen into something that plants can use. So the plants get the nitrogen from these bacteria, and then we get the nitrogen from those plants. So without the bacteria in the soil doing that, we would not be able to get the nitrogen that we need to survive, which is kind of crazy. And even in some cases, the plants kind of won't grow without these bacteria there, which is crazy. Like the plants need this bacteria to even grow. We do know that bacteria can help break down food, right? So if we didn't decompose or didn't break down dead things, I shouldn't say we. If nature didn't break down dead things, then all those nutrients would just get locked away inside of dead things. But these bacteria help break stuff down, and redistribute it back into the ground, redistribute it back into the plants and stuff so that other things can use those nutrients in order to survive. Inside of water, autotrophic bacteria are usually the bottom of the food chain, which means these little dudes hanging out in the water making their own food are usually the first things to get eaten. So the bacteria gets eaten by something a little bigger, which gets eaten by something a little bigger, which gets eaten by a fish, which gets eaten by a shark, which gets, like it just keeps going on and on. The basis for the food chain in the water is bacteria. If any of you guys like cheese or yogurt or vinegar, all that stuff is made by bacteria. We see here bacteria not only gives Swiss cheese its flavor, but also the holes, because it produces carbon dioxide that bubbles inside of the cheese. Yogurt has a bunch of bacteria in it. They're usually good yogurt. You can see commercials based off of like, well, this bacteria is even more beneficial for you. Maybe, maybe not, but the important thing to realize is that there is bacteria in yogurt. A lot of antibacterial medications that we have, antibiotics, are made from other types of bacteria, so kind of ironic. We use one bacteria to kill other bacteria. But again, that's incredibly important because there's some bacteria that are really dangerous to us. And if we can kill them, then that's good. Of course, as we've said, bacteria can cause bad diseases, stuff like that. Here's a couple of examples in case you didn't know. Strep throat, tuberculosis, tetanus. People think you get that from like rusty nails and stuff. Yeah, but that, not because the rust, because the bacteria chilling on the rusty nail in a puncture wound usually causes tetanus. This is not the rust, it's the stuff hanging out by the rust. That makes sense. Lyme disease, cavities. Oh my gosh, I hate cavities. I just went to the dentist right before all of these quarantine stuff and no cavities, so feeling good, feeling great. Actually, the dentist said, good job. I encourage you guys to brush your teeth if you don't. Diphtheria. Obviously, COVID-19 is a virus. It is not a bacteria, so none of that will be here. In the past, bacterial illnesses had a greater effect on human population than they do now. In the year 1900, your life expectancy was only 47. But we've pretty much increased that life expectancy to 75 years old. Mostly because, I mean, we know how to clean stuff. We know how to fight diseases. We've gotten better with antibiotics. Something kind of scary though, because we've been so good at killing off all the bacteria that can be killed off by antibiotics, in some cases, the only bacteria left are the ones that we can't kill with antibiotics. So if you guys can make that connection there, it's possible that in the future, there are some bacteria that we don't know how to kill. And this is already a problem we are seeing. We have to keep making new antibiotics because the bacteria that are more present are getting more resistant to everything we're throwing at it. So if that seems like something interesting, please go into the medical field and save the future of humanity to make sure that we can survive as long as possible. And yeah, that's it for bacteria. They're helpful. They're harmful. They can help us make food. They can help us make medicine. They can infect us and cause illnesses and death. They're chilling inside of your intestines right now, helping you digest your food and helping you absorb stuff. At the same time, your body's fighting some of them off to make sure you don't get any types of diseases. That's about a summary, so that's about it.